Can everyone see that okay?
Yep.
Cool, thank you. All right, so this is some summer work that I did with Dr. Nielsen. We were looking at identifying ortholog and paralog specific amino acids that were evolving from the highly conserved Drosophila melanogaster sperm specific beta-2 tubulin. And we were comparing its sequence to its major tubulin paralog beta-1, which is present anywhere else that sperm isn't, that tubulin's required in Drosophila, and its ortholog Glossina moristans beta-2 tubulin, which produces sperm and its relative tsetse flies. And we did this using molecular evolutionary genetics analysis software MegaX. So, all right. So kind of the big picture and some background with this. One of the big ideas that we have with this is that evolution is the result of selection due to advantages with no blueprint for the end goal. And what I kind of mean by this is just the fact that evolution doesn't have kind of an end blueprint for what's going to be good millions of years from now. It's just going off of what has the advantage today. So while evolution usually selects things that are advantageous to a species, it doesn't always, but it usually does. So when we think about what choices natural selection has to choose from, it kind of asks us what choices are available for various proteins. And we use beta-2 tubulin specifically to look at these choices that are available, found in the spermtail axoneme of Drosophila melanogaster as a model, because beta-2 tubulin and that structure are highly conserved and haven't evolved in over 60 million years, which kind of makes us ask why it hasn't evolved in that amount of time. And it leads to the question of whether beta-2 tubulin is the ideal configuration for the spermtail preserved by natural selection, and it continues to choose it, or if it's the only configuration that can support the lengthy Drosophila melanogaster spermtail axoneme, which is longer than human spermtail, and we'll go more into just how big it is here soon. Before taking on this bioinformatic approach of comparing sequences, we've done structure and function tests to check this with the paralog substitution of Drosophila beta-1. And we're currently in the process of checking this with the ortholog Glossina beta-2 that also produces sperm. And then those structure and function tests are just looking to see under the microscope if when substituted in our model, if they produce a motile spermtail. So during this last summer, due to constraints of meeting in person, we had to switch from these structure and function tests to a bioinformatic approach. So what we did was we aligned and analyzed the sequences between the paralogs and the orthologs to identify amino acids that are capable of evolving in these highly conserved proteins. So really quick, I'm just going to go through our research model just to give you guys perspective on what this bioinformatic research is looking at, like in vivo. So here's our flies, and you can see that they have various wing shapes or eye colors, and we use these different physical traits to help identify our male flies that are good for dissection to analyze these substituted tubulin proteins in order to identify which ones have the substituted sequences we're looking for. And this is just a photo of some of our flies in our lab from a few weeks ago. And then in order to get to the protein, we have to do test these dissections. And here's one I did under the microscope a few weeks ago, just to give you guys a perspective of what we're looking at here. And then we will take these testes and either look at them under the microscope to examine them and see if they're motile, or look at their cross-section to look at the microtubule arrangement. And then we'll also run gels to compare the size of these protein sequences when substituted. And then just for perspective, here's a cartoon of the spermtail. On the left, you have a kind of drawing of one, and then on the right, you have what the cross-section of it would look like. On the left here, I don't know if you guys can see my cursor, but I'm kind of like circling the spermtail. If this were to scale, it'd be about 10 times larger. Once again, it's just significantly large. And here you can see some other spermtail lengths below the drawing. And you can see that both flies are at least 20,000 microns. And then you can see humans at only 60. So you can just see just at how large these are when you think about how small a fruit fly is. It's kind of crazy how big their spermtails are. So it shows that might need something to pretty impressive to maintain that length. And then here's what the wild type beta-2 tubulin looks like. And this is the arrangement that hasn't evolved in over 60 million years. It has this 9-2 microtubule arrangement. And we refer to it as 9-2 because it has these 9 duplet alpha beta dimers in this ring here, and then 2 singlets in the center. And we'll look at that closer here on the next slide. So here we have our beta-1 on the left substituted in, and you'll notice that under the microscope, you can see that it just has those 9 dimer rings on the outside, but it doesn't have the 2 singlets on the inside. And then on the right, just for comparison, instead of a cartoon, this is what the beta-2 actually looks like under the microscope. And it's just kind of interesting because these proteins are more than 95% identical. So that 5%, so to say, has implications onto what the few critical differences are between these two that allow beta-2 to produce sperm and be motile. So next I'll be getting into the results I found from doing the alignments and comparing the differences between the sequences. So here's a photo of the paralog alignment and mega X. And you'll notice that I have 25 amino acid differences circled here. The red ones were considered based on their chemical characteristics, not characteristically different. So we didn't look at these site differences as closely, since alanine and glycine, for example, are not that different chemically speaking. We figured that the changes here wouldn't be as drastic as some of these blue sites that differ in chemical characteristics. And these chemical characteristics that I'm talking about when looking at these different amino acids and the different sequences comparatively, we based off of 2 criteria. The first criteria being if there was a difference in polarity between the original Drosophila beta-2 amino acid and whatever the substituted amino acid was, whether it was the paralog with Drosophila beta-1 and that last alignment, or what we're going to be looking at in a few slides with the ortholog with the Glossina beta-2. And then the other criteria we looked at was a difference in acidity between the Drosophila beta-2 amino acid and one of the substituted amino acids. And once again, there were 7 that were characteristically different in the paralog comparison, and there were 6 that were characteristically different in the ortholog comparison. So in order to locate and understand the interactions that were taking place at these characteristically different site differences, we used some papers from the Nogales lab, and specifically this alignment paired with some other figures in their papers to help identify whether it was in a beta sheet, helices, a loop, or any other protein motifs throughout the tubulin protein. And these papers were just super helpful in being able to identify and understand what interactions were occurring. And we'll kind of go into the interactions here. So between the paralog and ortholog, you can see here, there's only 2 amino acids where they were both different at the same site in the protein. And out of those 2 where they're different at the same site, there was only 1 where they had the same amino acid substituted. So this is just showing that there's not a lot of overlap between the paralog and ortholog, which shows that the evolution of these proteins isn't necessarily relaxed where they're both moving in the same direction. And then looking at the interactions that are occurring at these amino acid site differences, we have 3 longitudinal interactions here between these at these different protein motifs. And then we have 2 longitudinal interactions here on the ortholog. And these longitudinal interactions are between beta and beta across protofilaments of the protein. And then we have 2 lateral on the paralog and 1 lateral on the ortholog of these interactions. And those are just between the alpha and betas next to each other. But you can see here that all these amino acid differences are occurring at loops, sheet, and helices, which are all somewhat accessible and variable portions of the protein, so it makes sense that there's differences occurring at these sites. And then here's the ortholog alignment I was mentioning between the two sperm-producing sequences. You notice here instead of 25 differences between these, we have slightly less with 18 differences in total, 6 of which being characteristically different, and then the other 12 not being characteristically different. So then the conclusions that I was taken away from these results were that the differences between all 3 sequences show that there is flexibility in these conserved proteins, and it shows that they are able to evolve. And then also that the low overlap between Drosophila beta-1 and Glossina beta-2 show that evolution of tubulin isn't necessarily relaxed, but dependent on the need to manufacture sperm or not manufacture sperm. And then that the amino acids that evolved between beta-1 and beta-2 indicate differences in the needs of sperm, or being the major isoform and product of tubulin that's present and everywhere else other than the sperm. And then again, like I mentioned, most of these amino acids lie in variable regions like helices, sheets, and loops, with specific variable regions identified as, and then this is in order of most variation to least variation, with helix 3, sheet 4, helix 4, and sheet 6. And then the rest of the characteristically different variable regions were in unlabeled loops. And then the regions are evolving, are involved in mostly longitudinal beta to beta interactions across protofilaments, with just slightly less of these interactions being lateral alpha to beta interactions. And then the fact that the amino acids are evolving differently in Drosophila beta-2 and Glossina beta-2 indicate flexibility in sperm generation, and that there isn't just one cookie cutter way to get there, but there are different paths that they both can get there. Here's just my references with the Nogales paper at the top. Both search databases I use to get sequences, and then the link to the MegaX software, all of which are free if anyone's looking to using those for their research or anything. And there's my email if anyone feels more comfortable emailing me questions instead of asking me questions now. Thank you.
Hey, Colin, I have a question, maybe a half question, half comment. Could you share your screen again? Share your talk?
No problem.
And could you go back to the Glossina beta-2 alignment?
Yeah.
Wait, no, that's on me. There we go. So just to say, Colin, terrific job. So clearly presented, so well organized. Seeing your full talk and listening to you, one thing that's really interesting is these characteristically variable sites, the ones you circle in blue, right?
Yeah.
They're all found in the most conserved part of the protein, which is the middle part. I want to say kind of the late 100s amino acids to the 300 or so, 200.
Yeah, I got that like 120 to 140 region.
Yeah, that's a highly conserved part of the protein. It's interesting, not just amongst the orthologs, but amongst tubulins in general. And it's interesting to see that all the changes are occurring in this, highly conserved site or part of the protein. So we'll have to talk about that.
For sure.
Yeah. I think maybe you're off the hook.
Yeah.
Hey, but seriously, since you're there, how do you think you did?
I definitely went quicker than when I practiced it, which like happens with most presentations, but I liked it. It was nice. getting to look back at this work we did over the summer. It did take me a second to kind of like get back in the rhythm of like how we were thinking during the summer, since it's been a few months.
Well, you really did. You really did. I'm afraid I cut out, my internet lost you for a minute in the middle, but I heard most of it, and I think you nailed it, frankly. So and so far as the speed, you have a very calm, what do you want to say? Not just calm voice, but one that people have confidence in. You present in a manner that shows you understand this. That's really good for the audience.
Thank you.
I'll be in a little later, if maybe I'll see you. And yeah, congratulations. Real nice job.
Thank you. Yeah, that sounds good. I'll see you in the lab later.
Okay.
Cool.
Hey, take care.
You too.